the world of genetics entered a new era in 2012. Thanks to the discovery of a revolutionary tool, DNA manipulations are now more precise, easier, and more accessible than ever. The name of these genetic scissors is on everybody's lips: CRISPR Cas9. It enables to target, cut, and replace the genes of any living organism.
We can do the changing of the letters, or to induce mutations, or correct some errors in the original genome in such an efficient, rapid, and precise way.
Experiments that were only thought of before as something, oh, I would love to do that someday if I had the money and the energy and all that, become something that you can actually really plan for next week.
In just a few years, this discovery has totally transformed the outlook of research and its limits, and has thrown wide open the doors to the greatest hopes.
So having access to the DNA in the nucleus of the cell opens the door to a completely new curative approach. Go to where programs are directed, where mutations that cause disease lie.
I believe it will change how we do medicine in the future.
That is Star Trek, right? That's the fantasy of no more surgery, no more killing people through curing them with chemotherapy, but instead, when the doctor on Star Trek just gives you a little injection of something and your body rebuilds itself.
With CRISPR Cas9, the dream of repairing our genes, of curing our worst illnesses, is now within reach. This technology gives us enormous power of intervention, not only of our own species, but also over the planet's ecosystem. How far will we go? It is now time to find out what is incubating inside laboratory test tubes and to decide whether or not we want humanity to be tailor-made. The wind of change brought on by CRISPR Cas9 is blowing stronger on the west coast of the United States than anywhere else. World capitals of biotechnologies, the twin towns of Boston and Cambridge, are home to the biggest university research laboratories, the best hospitals, and the giants of the pharmaceutical industry. In the middle of this ecosystem, a medical startup with a difference was created in 2017. Founded by an association of patients suffering from Duchenne muscular dystrophy, which affects 250,000 people around the world, the young company is betting on CRISPR Cas9 to find a treatment for this incurable illness. Today, Jake Marazzo and his family are greeted by Megan Wood, representing the association Cure Duchenne. They want to better understand what the new therapeutic horizon opened up by CRISPR Cas9 could offer patients.
From what my mom explained to me, the technology helps stop the progression of Duchenne, but you just don't want this type of cure thing to have to wait to the technology of 3030. I don't know, I don't want it to be like far into the future where plate tectonics have moved.
I'm excited for all the new technology and the new hope, because without hope, what as a mother do I really have? When you hear Duchenne, it's pretty devastating to hear that your child has that, and that I actually started it and passed it on to him. So, my wish for Jake is that he wouldn't have Duchenne muscular dystrophy. So I pray that all of these new technologies like CRISPR hurries up. We need something faster. He's 14, so we need something faster. But we make the most of the life that we have.
Jake was diagnosed with Duchenne muscular dystrophy when he was 8. The illness first affects the lower limbs and then progressively reaches all the other muscles of the body, including respiratory and cardiac muscles. Sufferers rarely survive beyond the 30th year. This neuromuscular illness is hereditarily transmitted through the DNA. It's in the heart of cells that DNA can be found. All living beings follow the instructions it carries to develop, function, and reproduce. In the heart of a human cell, there are 46 DNA filaments condensed to the extreme. Each filament groups hundreds of segments of fundamental information, the genes. Seen up close, each filament forms a double helix, made up of 2 chains of molecules. The order in which these molecules follow each other, their sequence, is essential. A simple coding error on a gene can be enough to provoke serious pathologies. In the case of muscular dystrophy, the genetic defect prevents the production of a protein vital for the health of muscular fibers, dystrophin.
Cure Duchenne's mission is to fund or de-risk and vet early-stage science that will either treat or cure Duchenne muscular dystrophy. And we stumbled upon or vetted and found Dr. Olson's science using CRISPR technology. And from the early modeling, it's some of the most impressive science we've seen. So it was essentially a no-brainer for us to put forth a $5 million seed fund round utilizing our method of taking donor dollars and investing it in early-stage science.
Eric Olson directs a molecular biology laboratory at the University of Texas in Dallas. For the last 3 years, with CRISPR, he has been trying to repair the genetic errors that cause Duchenne muscular dystrophy in mice. The proof of the first successes of this research lies in these 2 images. On the left, heart muscle cells suffering from an insufficient production of dystrophin. On the right, the same cells that have been able to reconstruct after correction of the deficient gene. A complete repair that carries with it huge hopes.
With this new method, is it possible to allow me to become like everyone else to where I'm not?
But what I can tell you is that so far everything has worked better than we could have imagined. Certainly in the animals that we've now tested this in, we can restore not only the missing protein, but we can restore muscle strength in those animals.
Of course, our big concern are humans, so how long do you think before that can happen, trials in that area?
This is always the most difficult question, and I can't give you a specific answer on that. I can envision within a few years, if everything continues to go according to plan, that we could begin to think about moving this into patients. But we're talking years, we're not talking months, and we're not talking decades. Of course, you realize that this is the newest, most revolutionary technology in science, really, to be able to correct an error in the human genome. If you just think about that, it's really...
I know.
It's almost like science fiction.
It almost sounds that way.
To go back to the origins of this revolutionary discovery, we need to go to Berlin to meet Emmanuelle Charpentier. This French biologist made a name for herself around the world by revealing in 2012 the tool CRISPR Cas9. The publication of her article immediately rocked the scientific community. It's with the help of the American Jennifer Doudna that she managed her stroke of genius to appropriate a mechanism present in bacteria and turn it into genetic scissors within everyone's reach.
CRISPR is an immune system that exists in bacteria and that allows them to fight against viral infections. And this system consists of a small machinery that can be used like a genetic tool that makes it possible to modify the DNA of any cell using a molecular scissor system.
With its simple structure, CRISPR Cas9 can get into any cell quite easily and act with precision on the DNA. To identify its target, all you need to do is to program it with a copy of the sequence of the genetic code it has to work on. The tool will then be able to scan the entire DNA chain until it finds the corresponding portion of the gene. Then, the protein Cas9 takes over to separate and cut the 2 sections of DNA at the appropriate place. The gene is thus deactivated. On top of its search and cut functions, which are revolutionary in themselves, researchers are trying to improve the tool. By loading a spare segment of DNA onto CRISPR Cas9, it becomes possible in certain cases to obtain a pure genetic correction.
CRISPR technology can be compared to word processing software. That is to say, to recognize a word or letter in a text you want to modify, and to be able to use a simple system that allows you to specifically modify the word or the letter.
Just like software tools, CRISPR Cas9 is a breakthrough technology. Its applications are endless. Its developments are only just beginning.
If you want to genetic modify anything, in general use CRISPR because easier than... There are quite a number of other methods available. But no, everybody uses CRISPR because it's so much easier. So CRISPR has revolutionized how we do genetic manipulation.
There is clearly a before and an after CRISPR. In Cambridge, the director of the Whitehead Institute, Rudolf Jaenisch, is well placed to know it. He was the one who invented the first transgenic mouse in 1974 to study human illnesses.
In the days before CRISPR, you needed pretty sophisticated skills to make a mouse, a mutant mouse. You had to know how to treat embryonic stem cells or to select them, and they were delayed. It took us about a year to 2 years to make a mouse which carries this mutation. With CRISPR, what normally takes 2 years and a lot of skills takes now 3 weeks and not that many skills. It's maybe extreme when I say that, but any idiot can do it.
Even if the idiots in question often have a PhD, it's clear that with CRISPR, manipulating DNA has suddenly become infinitely more accessible. With genetically modified animals, practically mass-produced, the study of genes is moving forward in leaps and bounds. But even better still, the new tool makes it possible now to manipulate human cells directly, and thus help study phenomena that were inaccessible until now, like how our brain works.
This is very exciting. I think it's another tool to study complex human diseases.
Julien Muffat works under the direction of Rudolf Jaenisch. Strange living structures grow in his laboratory, such as mini brains.
The brain is a structured tissue which has layers, which has a cortex, which has many different subtypes of cells within it, and we can get some of that in those cultures.
These models obtained with stem cells are remarkable playing fields for CRISPR Cas9. For the first time, thanks to genetic scissors, degenerative illnesses like Alzheimer's or Parkinson's can be studied from a totally new angle.
You can now use CRISPR, for example, to mutate different genes and ask what does it do to the normal development of these mini brains. You can, of course, also ask, am I observing something that is relevant for Alzheimer's disease? And can I reverse or modulate it in a way that is therapeutically interesting? We're not creating gene therapies or cell therapies for the patient, but it's always in the back of our mind that that's something that could be used.
Thanks to CRISPR Cas9, our understanding of the living is accelerating. Prospects of therapies are getting closer. For certain illnesses like Alzheimer's, it's betting on the future. For others, the bets are on now. One of the biggest illnesses of our time is at the top of CRISPR's target list: cancer. In New York, at the Memorial Sloan Kettering Cancer Center, a number of teams are developing a new generation of treatment against cancer. The strategy consists in directly rearming our immune system. In particular, our white blood cells, called T lymphocytes, that defend our organism constantly against foreign or dangerous cells.
But when it comes to cancer, the cancer originates in our own tissues, and the immune system is not optimized to attack our own cells. In cancer, what we hope to do is to shape the T cells of the immune system to really fight and eliminate cancer.
With CRISPR, researchers modify white blood cells to help them recognize, target, and destroy cancerous cells as efficiently as they would a virus. Treatment begins with the blood sample. Cells are then selected, modified, and re-injected to cure.
What is very unique about this medicine is that it is based not on a chemical, not on a protein, like virtually all medicines today, but on a cell. It is what we called, a few years back, a living drug.
Even if this strategy has been followed for the last 10 years or so, CRISPR makes it more efficient, more accessible, and simpler. The first clinical trials have already begun.
And we never thought that a technology allowing for a very precise modification of the human genome would become available so soon. And now, it's here.
Another major illness could well be cured by modifying our immune system with CRISPR: AIDS.
HIV infection is devastating for people. The virus infects certain immune cells and kills them, and that leads to this disease. Now, the virus is known to need a certain receptor to get into the cells. And it's known when you destroy this receptor by a mutation, the virus cannot infect anymore. And if you now take these mutated stem cells, put them back into the patient, these cells will be resistant to HIV infection and basically form normal cells which can cure the patients.
CRISPR Cas9 has indeed opened up the era of the state-of-the-art DNA surgery. Could it cure everything? At the center of everyone's attention, the Swiss penknife of genetics is billboarded everywhere. The media tells of its latest achievements and wonders if the invention of CRISPR has already changed nature and affected the future of mankind. The excitement is also financial. As the list of diseases that could be eradicated by CRISPR gets longer, the amounts invested in startups and research are skyrocketing. In this expectation-rich phase, some remain cautious, others venture into the possibility of a disease-free world. Professor at Harvard University, George Church is one of the most renowned and provocative geneticists in the world. A pioneer of sequencing, the darling of the media because of his look, his avant-gardist and outspoken views, he has also taken part in the development of the CRISPR Cas9 technology. Thanks to genetic scissors, he's thinking about attacking the most inexorable and complex mechanism of our organism: aging.
90% of whom die of diseases that do not affect young people. So there's diseases of aging, and they are due to programming in our body to naturally decay and age. So some animals live 200 years, and some live 2 years on average, and that's because of their DNA. And so we're working on ways to do reversal of aging in my laboratory. You can take a very old cell and make it behave like an embryonic cell. That's how far you can reverse a cell.
So it's a real rejuvenating cure that George Church says he's ready to develop. Working with him, Bobby Dadwa patiently goes back to the origins of our cells.
It's quite exciting how we're doing this. We're not doing this work in animals. We're not doing this work in smaller organisms like mice, worms, or fruit flies, where a lot of aging research is being done. We're doing this in human cells. From one patient, I have samples taken 20 years apart. What I'm doing is I'm looking at their older self and seeing what can I do to that cell to make it young again. Your genetics are the same as if you were 90 years old to when you were 20 years old. The difference is how your genes are regulated. So we can turn on master regulators using CRISPR technology and take a look at which factors are very important to reverse aging. Often when you talk about to people about living longer, especially when I speak to my parents, my mother in particular, she doesn't want to live a long time because she doesn't want to be able to live in her hundreds and not take care of herself. So it's really important to mention that it's not just living longer, but living healthier, longer, healthier lives.
To regenerate us. That's the objective that Professor Church and his collaborators seem to be pursuing. And to offer an absolutely brand new future, he's using CRISPR Cas9 to solve a very concrete problem: the shortage of organs for people needing transplants. Following the early works conducted in Professor Church's laboratory, the young researcher Luhan Yang has launched a startup that aims to develop the marketing of genetically modified pig organs. It raised $38 million at its launch.
So eGenesis was founded in 2015 by Dr. George Church and me. And the mission of eGenesis is to create a world where there is no organ shortage. There are millions of patients worldwide are waiting for organ, and there is no medical solution.
But Luhan Yang is betting on launching again a solution left by the wayside a long time ago: xenotransplantation. The principle, using animal organs as an alternative resource to counter the lack of organ donations.
People attempted the concept of xenotransplantation for decades. Hasn't been successful due to the two technical hurdles, the immune compatibility issue as well as viral rejection issue. That's why the industry has abandoned xenotransplantation, and that's where we think CRISPR-Cas and eGenesis can come to play.
The pig is one of the best candidates to supply us with spare parts. It has organs that are very similar in size to ours and is biologically close to humans. But it is also a carrier of a number of inoffensive viruses for itself, but that could infect man. A problem Luhan Yang solved with a snip of genetic scissors.
The beauty of CRISPR compared with prior CRISPR tool is a multiplexability. By adding one Cas9 multiple guide RNA, we can tackle multiple sites in the genome at the same time. In 2015, our team member published another landmark study showing that we can eradicate all the 62 copies of virus from the pig genome. And the field got very excited. For one thing, we broke the record how many modifications we can do.
From their first laboratory successes, George Church and Luhan Yang have moved up to the next level, creating a breed of genetically modified pigs. It's in a breeding farm like this one in China that the first piglets were born in 2017. The babies are in perfect health. Here, in an annex of the Institute of Biomedicine of Canton, identical works to those of eGenesis are being conducted. This animal husbandry is not destined for food consumption, but well and truly to creating a porcine breed adapted to the production of kidneys, lungs, or hearts for our species.
Thanks to genome editing techniques like CRISPR-Cas9, combined with cloning techniques, we have been able to eliminate genes that cause problems with pigs and to add genes that were missing. We hope to be able to make it possible to transplant pig organs to humans.
To achieve total compatibility, eGenesis' challenge is now to introduce within the pig's genome genes that bring it closer to us. In other words, to humanize the pig.
There are different kind of modifications we are pursuing to mimic the human immune system, and once we know the efficacy and safety, we can potentially put that into the patient.
The humanization of animal organs can be widespread, just like launching a new production line onto the market. The organs from the same genetically modified pig could thus be attributed to different patients. In China today, our research is mainly conducted towards producing something useful. It matches public opinion demands.
Main partner, but also first competitor of the United States as far as these innovative research are concerned, China is determined to become the world leader in the sciences of the living and biotechnologies, and well intends on being taken seriously.
You need to understand that today China invests as much in research as Europe does, and they've decided to become competitive in relation to the Americans on questions of the life sciences, because the health market will represent 20% of GNPs of developed countries by 2025. So it's not by selling cars that the Chinese think they'll manage to achieve that deadline, but by selling medicine or health products like the Americans.
A vibrant showcase of this takeoff, Shenzhen boasts one of the highest growths in the world. Alongside state-of-the-art industries in the fields of energy and computing, biotechnologies have taken up a crucial position. Born in the late 90s to take part in the great adventure of decoding the human genome, the firm BGI has become, in under 20 years, the most important sequencing center in the world. The entire planet sends its DNA samples to decode all the genetic information they contain. Bolstered by this unique firepower, in 2016 the Chinese government decided to launch a colossal project, the China National Gene Bank, a true genetic Noah's Ark. It asked the BGI and its flamboyant president to supervise the project.
My colleagues and I have a dream, actually, which is composed of two parts. First, to sequence everybody in the world. Then the second dream, or the second part of that dream, is to sequence everything on the planet, on Earth.
No sooner said that it was almost done. The China National Gene Bank came out of the ground in 2016. The gigantic editing platform, spreading over 47,500 square meters, is there to transform the massive sequencing data into lucrative medical benefits.
If we should like to know why a human is a human, if we try to know where we are from, it's all related to our genome. So to know the genome sequence is essential. That's just the beginning to get that the book of life. Now with the book, of course, that's a long, long way to really be able to read it, to understand it.
In this room, each machine is capable of sequencing about 50,000 human genomes a year. To decode the human genome in 2003, it would have taken 13 years and $3 billion. Here, it is done in a few days for under $1,000. Carefully stored at -80 degrees Celsius, the gene bank already withholds the largest number of plant and animal genetic heritage in the world, of which 70% of all agricultural plants. An army of researchers and laboratory workers whose average age is 27 works hard every day at deciphering, understanding, recopying and modifying with CRISPR the genetic material at their disposal. To decode the great book of life, to care for and create new species. All this data is stored in a supercomputer of 60 petaoctets, the equivalent digital storage contained in 120 million films. The compilation, analysis, and interpretation of the data issued by these machines represents a real scientific and commercial treasure.
Why we should know more about the living things? Why should we try to change it? Now, the first is no doubt diseases, especially both genetic diseases and infectious diseases.
To know, target, and cure everything. That's the ambition that drives all the new heroes of genetics. In an age where sequencing and targeted modifications of the human genome open up inroads that were inaccessible till now. So, why wait for illnesses to develop before trying to cure them? Why not treat evil at its roots? The adult human body contains 100,000 billion cells, each with a copy of DNA. To cure a disease by genetic modification, you need to target every cell of the concerned organ. Even with CRISPR, it's a technically complex challenge with risks of error or unexpected reactions. The temptation to correct a problem by modifying DNA is great when it is contained in just one cell, the embryo. That step was taken in Canton in 2015. Less than 3 years after the invention of CRISPR, a team announced it had proceeded to a modification on a human embryo. As the international community was still debating the questions such an event raises, a new article came out in 2016. Dr. Yong Fan, a researcher in one of the biggest university hospitals in Canton, revealed his own attempt of intervening on a fertilized human egg.
Of course, we are aware of the ethical questions that could be raised. People told us, "You scientists are going to make designer babies, aren't you?" But that has nothing to do with reality. We work in a research laboratory in a hospital context. We scrupulously respect the laws of this country and the common deontology of all scientific research. So the public must not be scared of our work. For example, in China, we have a frequent form of a hereditary blood illness called beta thalassemia. That is why Chinese researchers are working hard on this illness. In the Guangdong region, it affects 13% of the population, or over 10 million people. Today, people can only be treated after the onset of the illness. What we're trying to do is to prevent the illness at the gene level before birth. It is our duty to take care of people's suffering and to find radical solutions that cure. That is why we are conducting research on human embryos to find ways of eradicating the illness on a genetic level. Thanks to in vitro fertilization techniques, we will be able to eliminate the disease, and it will be a huge step forward in public health and in the quality of life of Chinese populations.
The prospect is dizzying. To manipulate the DNA of an embryo is creating a genetically modified human right down to its reproductive cells and brings a mutation that could be transmitted to the following generations. Even though we are still at the stage of fundamental research, with embryos destroyed after a few days, the announcement of the first works undertaken by the Chinese on a future baby has set off an uproar within the scientific community.
Should we do that? Should we think about changing the human genome for the next generations?
Faced with the given fact, it is high time to ask a few questions. However, it seems a little late now to press the pause button.
The moment we realized we had been overrun by the turn of things, we came to the obvious conclusion in the autumn of 2015 that it was impossible to stop the machine. The idea that we could have put all the people capable of using the CRISPR technique in one place and asked them to stop for a year immediately seemed impossible to everyone.
Is it really possible to harmonize the policies and laws all over the world? No. Is it possible to enforce laws all over the world? No. And is it possible to make sure there will never be some rogue agent who goes forward and does experiments we think are unethical or unsafe? No. But we can minimize that possibility.
The electroshock provoked by the Chinese works on embryos pushes scientists to react. Late 2015, the Chinese and American Academies of Science called a big summit together. The question of where scientific research should stop is raised because in both leading countries, no legislation forbids to run research on embryos, nor prohibits in principle the birth of a genetically modified baby.
Around the question of the embryo, it is clear we are in a legal Tower of Babel. There are countries with total prohibition and countries with total liberalism.
The summit in 2015 was really the result of concern by scientists how the CRISPR technology would be used to human application, to propose guidelines under what circumstances one would prevent or permit the genetic manipulation of human embryos.
One part of the initiative was to see if we could develop some harmonization around the world in national policies about what should be done, what might be done, and what should never be done.
At the end of long discussions, scientists appear to be unanimous. Research must go on. The opportunity of one day being able to eradicate a disease at the embryo stage is too good to let go.
It is not intrinsically unethical to imagine using it for so-called germline editing, which would change your children. That's not to say you should do it, but it does suggest that it's a choice that governments can make. It's not something we view every government must prohibit.
You cannot turn the clock back. We cannot say, hey, let's stop. I mean, a scientist has the freedom to explore the unknown, right? We recommend even reproductive gene editing could be allowed if there's no treatment alternative for the conditions under study. But whether that technology will be harmful or beneficial, and that had to be a societal decision. It cannot be a scientific decision.
By allowing the research on the modification of embryos to go ahead, the scientists gathered in 2015 are well aware of one thing. Their decision concerns the whole planet. The stakes are written black on white in the final conclusions of this summit. Once introduced into the human population, genetic alterations would be difficult to remove and would not remain within any single community or country.
I don't think it should be only in the hands of the science community, nor should we wait until 7.5 billion people weigh in on it. It's something in between where you want to get as many people as possible engaged in the dialogue, as many different kinds of points of view, but you're not going to get it to everybody, but we should certainly try.
The CRISPR revolution arrived so quickly that it took everyone by surprise. As a result, a new kind of popular science wants to draw citizens into the debate by opening up research to non-academics. Brooklyn, an emblematic New York district. This is where, 10 years ago, the first community lab opened on the fringes of the academic world.
You just kind of want to get any cells that might be ready to fall off. And then open this, and you're going to swish it around. So that's about enough.
In these open and participative facilities, amateur biologists or just ordinary science enthusiasts are encouraged to mingle and discuss what is at stake.
You see the pellet at the bottom right there? Wow. The white thing? So see if you've got one. This is where we find out if you're a robot or not. A community lab is a laboratory that exists independently of another organization, and that is a grassroots effort by and for the community. That means that you can study whatever you want. You don't have to study something that is fundable. You can work on something that's never going to make any money, but it might be really valuable for mankind or even just for your neighborhood. So when we first started the citizen science movement into community labs, CRISPR didn't exist. We started thinking about having CRISPR classes. We weren't sure we wanted to have them because we kept being approached by people in the press who wanted to make a big story out of how scary it was that citizen scientists were using CRISPR. In the end, we decided that we would teach workshops and classes to the general public, but we would be the responsible CRISPR people. When we teach classes in these community labs, we try to put the technology in context. So we talk about the controversy associated with germline editing. We talk about how people have very different views on whether or not the human genome should be edited. That's something that I think the people that come into the classes in these community labs love to discuss. They're there because they're interested in what this technology can do. They're curious about it, and they want to enter the discussion.
Scientists have the obligation to explain what they're doing to the public. What are the potential, what are the dangers of the science people do. And I think there's a lot of misconceptions about science. So I think there's a need to explain the science to the legislators as well as to the public, to really inform people.
For public debate to happen, scientists will have to answer the fundamental question about security. For the time being, trials mainly end up failing, but research is intensifying and sooner or later we'll get there.
Without banning the idea of one day curing embryos, we need to give ourselves the means to do it in conditions of security and efficiency that will allow us to turn it into a treatment and not just a game for laboratories. It needs to be 100% solid, and it's got to work every time.
Any change conducted on an embryo's DNA, whether beneficial or accidental, will be transmitted to future generations. We shudder in anticipation. What threshold of security will be set? And even if one day a complete guarantee is given, the debate will continue because the technique leads to another perspective, the improvement of the human being.
That opens the door to what should we edit. As it becomes more commonplace, then you wonder where it's going to stop, where along the continuum do you go from the necessary to something that you want, to something that is desirable that may not have any health effects one way or the other. So there's always, when you talk about editing human embryos, there's always the specter of eugenics on the horizon.
Improve an individual by what's called enhancement. For example, to make them taller or more strong or more intelligent or blue eyes, for example, right? And the guidelines say, no, this is not permissible.
The fantasy that we can enhance ourselves with this is overblown. We can't define intelligence, let alone change it. Even your height, which seems very straightforward, is the result of literally hundreds of different genes as well as your environment.
In some cases, the technology may be slow to appear or never appear, and so the discussion might have been wasted. But it's better to waste a discussion on an important ethical topic than to miss it completely and have things come up too fast.
CRISPR is pushing us to reflect on what we want individually and collectively. But genetic scissors have an impact far beyond our own species. We're now looking into modifying entire ecosystems to combat some of our illnesses. In Terni, in the center of Italy, the Pole for Genetic Innovation houses a high-security installation. In this ecological confinement laboratory, climatic chambers have been designed to reproduce the habitat of the African Anopheles gambiae. This species is the most important vector of malaria that kills almost 450,000 people every year. The experimentation that is being carried out here aims to test a radical strategy, the complete eradication of the species. In researchers' heads, the equation is simple. Without mosquitoes to transmit it, the illness could be wiped out in a few years.
All available technology, in addition to insecticides, for example, draining swamps or managing the environment such that you're reducing the breeding size, were beyond the capability of resource-poor country. This measure requires a lot of money. So we thought, well, if we develop a genetically modified mosquito or we reduce their reproduction ability, then we overcome the major problem, which is sustainability. Although this genetic modification cannot be transmitted, you have to release 10 times as many mosquitoes there are on the field, which is absolutely not feasible, unless we have developed a system that is able to overcome the law of heredity and spread the genetic modification. And this is where gene drive comes into play.
Gene drive is a modification programmed to speed spread across a whole population. In normal times, a gene carried by a parent only has a 50-50 chance of being transmitted. Thanks to gene drive, as soon as a modified individual reproduces, it imposes its tampered gene to all its descendants. The strategy is akin to hijacking because it works a bit like a Trojan horse, where the modified gene itself contains the necessary material and instructions to transmit CRISPR and to duplicate the modification indefinitely.
In this way, at every generation, like a domino effect, the genetic modification spreads. We introduce CRISPR in such a way that progressively destroy genes which are important for mosquito reproduction. So the end result would be a population collapse.
Gene drive provides a novel and powerful means of action. Its potential to eradicate an illness is enormous. So are the environmental risks.
On one hand, you have hundreds of thousands of human lives that can be spared. 438,000 deaths last year. And on the other, a potential impact on biodiversity that we may not necessarily be able to stop, like a chain reaction of sorts that we'll have provoked. And here we bring up a whole ethical line of questioning, a whole series of needs for answers to a vast number of questions linked to human health that have an impact on different environmental elements.
In terms of just risk, of course, the ability to model the ecosystem, to really know what the consequences are of your environmental intervention is very, very limited. It's a complicated system with lots of consequences that spill out over years.
So how do we feel about that? And how much information should we have before we actually do something like that as a society? Because we don't know what impact it's going to have on the ecosystem, not really.
We will be able to provide a lot of information in terms of, for example, how long the genetic modification will take to spread. We will be able to say whether it spreads to other species which are very close. We'll be able to check if you can detect trace of DNA in the predators, to assess ecological parameter, assess the safety, produce material then for convincing people to move to phase 3, which will be a confined release in the field. And then from there to open field release.
The sequel to this story has not been written yet. How will we be able to find ways of studying in even finer detail the possible consequences before proceeding to full-scale testing? And who will give those tests the go-ahead?
It is urgent that the decisions be not limited to just a few laboratories in developed countries, but that other populations be involved, because where are we going to be doing these manipulations? They'll be done in Africa, or they'll be done in Southeast Asia. And it might be up to the Africans of Cameroon, Gabon, or the Vietnamese to choose whether they want to test these kind of strategies with the promise of getting rid of malaria, for example. But today, that's still just a promise.
Now, we, in principle, need to achieve a point where people regard this technology as safe and they are prepared to accept it. And then we need to build an international framework by which every country accepts that the neighboring country is prepared to release the gene drive. Because unfortunately, then the technology will not recognize political boundaries.
CRISPR opens the way to true planetary bioengineering. The cards have been reshuffled, so to speak. All fields are concerned. Because of the extraordinary prospects for human health now within reach, we have almost lost sight of another major field of application for these new techniques: agriculture. And once again, the new tools of genetics change the game radically by rendering the actual regulations on GMOs null and void.
Today, the definition of a GMO is a plant in which we have introduced a foreign gene. Therefore, we have introduced a foreign gene to give it a specific feature. For example, to resist to a particular bacteria, or to resist a virus, or resist a fungus. Today we can do the same thing without introducing a foreign gene and without leaving traces. So we are starting to have plants that have undergone genetic modification. They are therefore genetically modified by engineering the genome. We call them MOGEs. But they no longer correspond to the definition of GMOs.
One of the test cases for this in the United States was an investigator at a university created a mushroom that was genetically modified with CRISPR not to turn brown on the shelves when it got bruised, which of course would be a great thing for the produce department in the supermarket because you could keep those mushrooms there forever. And he actually got an opinion from the FDA, and they said, no, by our definition, this is not genetically modified because it doesn't contain any foreign DNA.
Currently in Europe, there's a large debate going on about whether or not editing is the same thing as engineering when it comes to foods. It's possible that Europe may soon see more edited foods, even though they don't allow for the sale of genetically modified foods.
Here again, the controversy is at its height, fueled by enormous sanitary, ethical, and financial stakes. Whether it concerns our health or our food, CRISPR pushes back our capacities to modify any living organism and raises as many hopes as it does concerns. We don't need to choose between the two extremes, but rather to think things out.